emergent variants and trying to understand how they might change the spike's behavior and the efficacy of vaccines, for example, and inform the design of new vaccines. As I said, we've been doing the same sorts of things with every other protein from the virus we can. All the data is available on AWS and the OSF if you're interested in playing with it, and you can see our bioRxiv paper for the links. With that, I'm extremely grateful for the Folding at Home team, both the scientists spread around the globe and also our volunteers who are even more globally distributed, and all of our funding sources, including the NSF, for their help keeping this going as we struggled at the beginning to scale up in response to the massive increase in participation in this project. Thanks.
Thanks very much, Gregory. Really exciting to see how much interest there is and really fascinating talk. So thank you.
Thanks.
Our next speaker is Michael Kinzel of the University of Central Florida, who will be discussing different mechanisms of airborne pathogen transmission of the virus. So Mike, whenever you're ready, please feel free to go ahead.
Okay, do you hear me?
Yes, we can.
All right, well, yeah, this talk is on essentially studying fluid dynamic drivers associated with pathogen transmission. It's done by myself, focusing on the computation, and Kofi Kareem Ahmed doing focusing on experiments. We have a pretty nice team of postdocs and PhD students that have been helping us out on this effort. Okay, here we are. So ultimately, we're asking if we can control transmissibility by controlling basically the underlying fluid dynamics in your saliva. It basically comes down to what we see here is called the Wells curve. What you can see here is droplet size. These are droplets emitted through speech, coughing, sneezing, these sorts of things. And this is time. And what happens is for very large droplets, they tend to fall to the ground. So this is a time that it takes a droplet to fall 2 meters to the ground. But at some point, it evaporates faster than it falls to the ground, and it becomes this airborne type path or an aerosol. So how does this relate to the different transmission paths? Very large droplets, they tend to just fall down to the ground, and they actually don't, they're very hard to basically breathe in. They don't really drive social distancing as much as the mid-range ones. These mid-range particles are the ones that get trapped into the air that you emit as you speak, cough, and sneeze. And those are the ones that travel far. And then we have the airborne paths. So this is the ones that evaporate and they just circulate throughout the room as very tiny particles. What generally happens is the droplet evaporates, leaving essentially a viral particle with a small amount of, or that's still survivable and can transmit the illness, but it is not necessarily, it is very small and just floats around in the air essentially. So the goal that we're trying to focus on is, can we control the underlying fluid dynamics associated with the generation of these droplets that generate on average larger, heavier droplets that have a tendency to reduce the transmissibility. So we want to make these droplets fall rather than spread out around the room. So the basic concepts we're studying are viscosity, so how thick it is. You can see here the honey, it's very thick fluid. You can think about variations in viscosity in your mouth, and specifically in saliva and mucus, and as well as content, how much saliva you have in your mouth or mucus in the various films in your body, and how that may relate to transmissibility. We're looking specifically at direct transmission using numerical simulations and experiments, as well as airborne transmission, specifically in rooms. So we're looking at things like restaurants, classrooms, these sorts of things. And ultimately what we're trying to ask is, how do saliva properties, viscosity and content, relate to social distancing and capacity of rooms? Can we study this to better understand what's driving super spreaders? And can we develop products that can actually reduce transmission based on things other than a mask? So this is some results from our numerical simulations. This is a thin saliva. A way you could conceptually see that is just a spray bottle with water, right? It atomizes, or it breaks up into a lot of droplets, and it basically spreads throughout a room, just like, and this is what we're seeing in our simulations. These small droplets tend to float, loft, and aerosolize. On the other hand, you can think of a very thick fluid. A very easy way to think of this is this, I can't believe it's not butter spray. It just falls, right? It leads to very large droplets. And when we study this, we are seeing and observing that, yeah, these things, droplets don't fall. So these are all human sneezes, and you can see that the majority of the droplets, the majority of the content of something that is emitted, or all the fluids that are emitted during a sneeze, have a tendency to fall to the ground. So from a probability standpoint, this is the more ideal scenario, because it's going to be less likely to transmit the virus. If we kind of summarize this is kind of a code plot after about 5 seconds. You can see these red droplets, they're all the thin saliva, they're lofted up in the air, they're not really falling too much. The thicker droplets are tending to fall. If we look at essentially droplet counts, so number of droplets as a function of droplet diameter, this is all within kind of like a region around here where things are more susceptible to transmission. You see you have a lot of thin or the thin saliva leads to a lot of droplets that are lofted and very prone to transmitting coronavirus. Versus when you have thick droplets, you have very few of them, they're very heavy, and they tend to be much larger, they're gonna end up falling. This is kind of one of the, some of the findings from the study, but we're finding a lot of other things too. When you dig into the dental literature, you see that there's a lot of tendencies for humans to actually have thicker saliva. So somebody who is older, stressed or ill, or even women as compared to men, they all have thicker and less saliva, and they're going to be less likely to be transmitting disease. So kind of our ideal super spreader is probably a younger male, 18-year-old kind of man is probably the super spreader profile. But we also wonder, this does pose a question: do humans actually naturally respond to reduce these airborne transmission routes? It may be it's an interesting question that, at least it seems our initial studies are pointing to maybe they do. We also had found that if you're congested, your sneezes will travel about 65% further, basically blocks up the nasal flow path, leading to a stronger jet that comes out of your mouth. If you're interested, we have a physics of fluids paper that kind of highlights some of our results. The other thing we're studying is airborne transmission. We just have a paper out studying classroom safety. Basically, a few key results here are we're finding that using advanced modeling, we do see a pretty consistent comparison with kind of like the estimators that say how safe it is for you to stay in a room with a certain number of infected people. And the worst case scenario we found was those numbers are twice as bad as the worst case scenario of all the different transmission routes. So you can think of this classroom where you have 9 students, 1 teacher, there are a whole bunch of different transmission routes. And we were trying to identify the worst case transmission route. And that case was only twice as bad as the estimators, which is, I think, a good thing to find. Another thing we're studying is the effective ventilation system. This is a room with ventilation. So this is kind of a distribution of all the transmission routes. And when we have this distribution, we see about a 2 to 3% transmission rate under a lot of assumptions. Under the same assumptions, an unventilated room is about twice as bad. Last kind of result we see is infection probability in the context of airborne route is very weakly correlated to distance. So social distancing is not that effective when you actually are focusing on the airborne route. So where we're moving with this next is to study, most of these studies have been under the assumptions of mask. We want to look into the assumptions of our fluidic control type effect. We actually, we think we'll get about an 80% reduction in aerosol using fluidic control. So if we move just quickly into broader impact, we've been able to get some, to help actually Good Morning America a few times studying or, using our visualizations to help them show, safety, how safe are certain things. So this is one study where we were showing, hey, you have these barriers, they help, but they aren't a fail-safe. don't not use masks. And then we're moving to another Good Morning America piece with Martin Bezant at MIT. Here's actually a simulation of an entire church. And essentially, we're able to visualize how aerosols kind of move around in very big room environments. Okay, kind of the last thing we're looking at is how do we actually take this and implement this in reality. So we actually have developed a chocolate that does exactly what we're saying, makes thicker and less saliva. And we think that it'd be a nice way to, CDC is actually recommending now double masks, really tightly fitted. We think this might be another route, a single mask with a chocolate that is, much more desirable or much more acceptable to the public. So if you have any interest, you could check out we did a National Academies I-Core and pitch with them, and we have a website. We're trying to get this off. So hopefully this is another, we think we can take it to a solution. That's pretty much it. If you have any questions, feel free, I'll stay around.
Thanks very much, Mike. Really great to hear more about fluid dynamics after Howard Stone's talk earlier this fall. I love the solution created there. So yeah, I love Miranda's comment. We can all get behind chocolate. So our final speaker today is Paul Westerhoff of Arizona State University, who is going to be talking to us about disinfection and reuse of healthcare worker facial masks. So Paul, whenever you're ready, please go ahead.
Great, thanks. And Michael set us up pretty well thinking about these airborne particulates and then how do you get rid of them is to wear a mask. And so if we think back to the fall, we didn't have many of these masks. And so the kind of purpose of this study was to understand, the viability of reusing masks. So this is our group at ASU, both in engineering and chemistry. So I'm going to actually start with the broader impacts because I think as all of us have kind of alluded to, it's pretty easy to understand them over the last year or so. And really what this project allowed us to do is to have some core capabilities and flexibility to help people. So we were able to kind of aerosolize particles in a nebulizer, and then we could put in different mask materials kind of right in this interface. And we could measure particle size distributions in the air kind of before and after this mask to understand the efficiency of different masks. We think back to the spring, there were lots of people, saying, what about using used t-shirts and other things as mask materials? So our team have really helped a lot of different groups locally kind of understand the viability of cloth masks and other things. Really cool, our team was able to provide data for the student team at ASU, which actually won the X Prize competition for the next generation mask. They won $500,000. And so they were able to use data from Pierre's lab in terms of looking at this filtration efficiency during their pitch to show that their masks actually functioned. So congratulations to that team. Also, what we were looking at was how to help first responders. So these first responders back in the spring didn't have enough masks or other PPE. They didn't want to treat them with chemicals because it's hot in Arizona. They didn't want to put wet masks on them. They really wanted to be able to treat them in between break sessions at, in this case, a firehouse. So after several iterations, we came up with a design that these firemen are showing here that they actually deployed and used to disinfect their masks. And so there were UV lights on the top and bottom, kind of a grill in the middle, kind of like a barbecue. And within about 30 seconds to 1 minute, they could disinfect their masks and reuse them again. So we did, of course, more scientific, studies here as well in terms of answering the basic question, not only can you reuse masks, but do applying UV light, kind of a non-chemical way to use these masks, damage the polymers that are responsible for removing these airborne viruses. So we looked at both low pressure lamps and LEDs. It actually turned out in the spring and summer, it was really difficult to purchase some of these. And so there was a real rush on this. So we did look at focusing on these UV lamps. Here's an example. We're able to disinfect, these masks using 1 joule per square centimeter. So, obviously about 10 times higher than we really needed. Then we went all the way up to 10 joules per square centimeter. That would show multiple recycling efforts. And essentially in all cases, we're able to remove greater than 95% of these airborne particles. So clearly UV light, can be used to treat these masks. We went deeper into understanding the polymer chemistry of these masks. There are multiple layers. We looked at irradiating each layer at pretty high UV doses and really looked at the fundamental chemistry and showed that the UV light really would not change the chemistry of these fibers. These are not small pores that remove particles by size exclusion. They're pretty big because you have to be able to push your air through them. And so it actually becomes the electrostatic interactions. We're able to, kind of continue this, look at different types of masks as well. Finally, I want to say, kind of where are we going to kind of keep us on time here, but we're interested in particles in air that have different charge and masks that have different charges. So it turns out that when you look at these masks, again, you can move air through them, so they're fairly porous, but it's a charge on the mask. And so we've been measuring the electrostatic charge of these mask materials to see how they interact with positively or negatively charged particles in the air. And this to give you some idea of the relative importance of the charge on the membranes in these filters. This red looks at very high removal efficiency of masks that as you purchase them and even after reuse. But if you remove the charge, you get really only about half the treatment efficiency. So the methods that you use to make the masks, but also to clean and recycle the masks are really important. And it turned out that UV light did not impair this electrostatic performance. Here I focused mostly on the mask, but we did a lot of work on light emitting diodes in the disinfection range, helping various startup companies bring products to market fairly quickly by looking at disinfection on surfaces as well. And again, we have publications kind of in process, but it was really kind of the fun part in our group to get the students in our labs kind of engaged in solving kind of a real world problem. We actually created a lot of good, synergy amongst our research students doing this work because it could really be used and helpful to the broader public. So with that, I'd like to thank NSF for their funding.
Thank you very much, Paul. Great talk, and really excited to hear both about the students' involvement and engagement and their great win there. On that note, let's switch over to the Q&A portion of the event. We've been having one of our students keep track of this in the chat window. Abhishek, were there any questions that you could share that have come from the participants?
Sure. Hi, Katie. Thanks. So there were quite a few questions that participants asked over the speeches. And so thankfully, our speakers have been quite active in answering most of them. There's one question, though, that hasn't been answered yet. And it's for you, Timothy, if you'd like to take it. It's, does your forecasting model take into account the vaccination rate as well?
No, at the time we built the model, there was no vaccine. So we did not take into account vaccine at the time we built the model. And another thing is right now we're working on the impact of COVID, the severity, and we try to map it to specific areas that are severe such that vaccine can go there as a priority or to unlike places where the impacts are not very felt. So when we built this model, the vaccine was not a factor.
Thank you so much for the detailed answer. I think that's about it. All questions have been answered from the ones that were asked in the chat. And some of the speakers were kind enough to share links to some of their written material as well. So I'd encourage the participants otherwise to, just go and have a look at all of that so that that might answer their questions even more. So.
Absolutely. We really do encourage folks to feel free to follow up with each other, particularly if you've heard from someone whose work might be alignment with yours in some way. We've had a number of great collaborations and other partnerships come out of these conversations that have started on the COVID Info Commons network and these events. So please do feel free to follow up as well as with the team here. If there are no further questions at this time, I'll just close by thanking everyone for joining us today, the speakers for their great presentations, all of you who have joined us to hear from them, and many thanks as always to NSF for their support both of these great research projects and of the COVID Information Commons itself. As you can see on the screen here, full information on all of our upcoming events and opportunities is available at covidinfocommons.net. There is a short link in the lower left to sign up for our mailing list to receive these updates in your inbox. It's bit.ly/kick-news. You can also access us from the main website above. And again, just as a reminder, if you are interested at all in learning more about the undergraduate student paper challenge that the COVID Information Commons is organizing this spring, or you think that you'd want to share this info with undergrads in your network, again, please feel free to visit the short link in the lower right there, bit.ly/kick-student-challenge, also available via the main website, covidinfocommons.net. I think that will be all for today. So thank you again to everyone for joining us, and hope you have a good rest of your